Okay, maybe we can get going. So yeah, welcome everyone. Thank you very much for attending this really important topic on international data exchange and data representation standards for metabolomics. And we're also including, other communities here, lipidomics and exposomics here. So we're actually really excited to share a new grant with you, a new consortium here. So the initial plan was to call it the MetabolomeXchange. You may know that this already exists in the public domain, but once the consortium got going, we realized we wanted to do a lot more than just simply aggregate data sets for the community. So we come up with a new name, the Metabolomics Hub. Again, this is a partnership between Metabolomics Workbench in the US, MetaboLights, EBI in the UK, and also GNPS Massive in the US too. And this is supported by the Chan Zuckerberg Initiative and also the Chan Zuckerberg BioHub San Francisco. So you'll hear a little bit more about this consortium during the session, and then we'll have some discussion throughout as well, where we are asking you to get engaged and to participate in this. So just a reminder of the workshops. We're here because we want to communicate and collate the need for the generalization of open data practices across metabolomics, and we'll hopefully show you some example efforts. We want to stimulate the engagement and energize the awareness of data standards, and hopefully we can start to think about a roadmap here, including the main challenges and opportunities for the future. So hopefully by the end of this, you will describe the benefits of widespread data, open data practices, and the role of the Metabolomics Hub project herein. Hopefully you can understand the current landscape of data repositories and data standards across the metabolomics, and then hopefully cite some examples of some minimum guidelines in the field as well, and efforts ongoing. So yeah, this is the agenda for the workshop. So we have Juan Antonio to kick off with the need for coordinated open data practices in metabolomics, and here specifically about introducing the Metabolomics Hub project. We have Rick Dunn here to talk about the metabolomics community requirements and demands and next steps. And we have Nils to talk about the extensible metadata standards for mass spectrometry of small molecules, and then Elliott here to talk about fair chemical exposome, similar needs to metabolomics, but the challenges amplified. And then we'll open it. So the idea will be that we will have 15 minutes presentations, and then after each presentation, we'll open up for audience participation for about 10 minutes or so, and then we'll have a panel discussion at the end with some 3 themes to get you guys to think about the future of the community. Oh yeah, so Juan, it's over to you, thanks.
Okay, so, no. Okay. No, this is very sensitive. That's it. Okay, hello everyone, and thanks a lot for joining the workshop today. So, my name is Juan Antonio Vizcaíno, and I work at the European Bioinformatics Institute in Cambridge, in the UK. I'm going to really give an introduction to what Metabolomics Hub is. So, this is the overview of my talk. Of course, feel free to ask any questions at the end, and in any case, we are going to be here around the whole week. So, hopefully, we'll have time to interact with many of you here. So I'm going to start with a short introduction about ourselves. Then I will talk about motivation and overall objectives of the Metabolomics Hub Consortium. I will explain the overall concept. I will give a primer about the importance of open data standards. And then I'll finish with just one slide devoted to conclusions. So Thomas here, but also Oscar and Noemi, we are based in the European Bioinformatics Institute in Cambridge, UK, which is the largest provider of biomolecular data worldwide, and we are responsible for MetaboLights as the open repository for metabolomics experiments. So this workshop is aimed at improving fair data practices in the field. So maybe I don't need to explain too much about what fair means, because this is really used more and more all the time, not only biology, but also for any type of data these days. So fair means fundable, accessible, interoperable, and reusable. In the context of making data fundable and accessible, data repositories and databases are really key. And this is really what the MetaboLights team has been doing for many years, since 2011, when the project was started by Christoph Steinbeck. I personally only joined last year this effort, and is an open repository for all types of metabolomics experiments. So MetaboLights uses the ISA data model and is very tightly connected with KEBI, which is the database at the EBI for small molecules. Okay, I also want to, since we are not talking here today about MetaboLights, but about Metabolomics Hub, I really want to highlight that our partners in this project are Metabolomics Workbench. Here, EOIN is in the audience, and also GNPS Massive, and I think Yasin is somewhere in the audience, although I cannot see him, but just to see that this is not only us, this is really a integrated team of 3 resources, and also others as well. Okay, so after this short introduction, I'm going to talk about the motivation and the overall objectives of the consortium. So this is really what is just a graphical representation. I'm trying to see how this works here. Okay. So of what we are trying to do. So at the top is metabolomics practitioners, you. Then we want really that the data gets submitted into one of the Metabolomics Hub data resources. Again, Metabolomics Workbench, MetaboLights, and GNPS. I will mention this later, but this is by no means a closed club. We are starting this because someone needs to start it. But the idea is that this will be open to any kind of other resources that are in, other resources that in the future. But, someone has to start the framework, someone has to start the work to enable this infrastructure. So is the part of the devoted to the data submission. This is a bit complicated, actually. And then there is the part related to data reuse. Maybe this is easy, actually, sorry. It's the part related to data reuse, and that's part of what is really ambition to benefit anyone in the community, both computational and data scientists, bioinformaticians, but also web lab scientists and biomedical experts. And the idea is that the open data with certain requirements gets reused more and more by itself, or also integrated with experimental data that is generated in the different labs. At the end, the objective is to have enriched, reusable, and publicly available process data that can also be AI ready. Okay, so the motivation of the consortium is really to standardize open data practice in the metabolomics and related fields. And by related fields, I mean especially lipidomics and exposomics that are also represented in this workshop. We really need to standardize as a field what is a data set in a public metabolomics data repository, which components, data types, and metadata should it have. And this needs to be standardized. Also, the consortium was to have a common voice to communicate with journals and funders in the context of open data practices. And if I had to choose one thing myself, is to, if we manage to do something, is to generalize the availability of metabolomics data sets and ideally other data types as spectral libraries in the public domain. Really, the objective is to promote increased data reuse for the benefit of the field. So the overall concept is what we want to do is to make life easier for you. So make life easier for everyone in the community by implementing the standard data submission and data dissemination practices between the main metabolomics data resources. Here, MetaboLights, Metabolomics Workbench, and GNPS. So, the process of submitting data to these resources should be very similar. You shouldn't really notice much difference. Of course, the interface can be different, but the requirements, the assumptions, the guidelines should be very similar. And this is what we want to implement. It should be a completely open framework, enable the other resources like MetaboBank or MBPost or the Korean, the Indian repository may join in the future. We want to support at this point open data. That means no human-sensitive data, because that brings a lot of extra complications due to control access. And this is not the kind of problem that needs to be solved in a first iteration of this consortium. So at the moment, we are going to focus on open data. And really the aim is to generalize and coordinate open data practices in the field. So this is the way it will work. So here is the metabolomics community. Data gets submitted to one of the Metabolomics Hub data resources, but also others in the future. The data set by default are private. At that point, the data can be shared with journal editors and reviewers. And at that point, hopefully, the paper will be published. And at that point, the data set will be made publicly available. And then all publicly available data sets will be used to populate what we call the Metabolomics Hub portal. That is basically a centralized place where everyone can access open data in the field. So data sets hopefully in the future, spectral libraries and other types of information. So there will not be the need to go to each individual resource, but there will be the portal who will integrate all the information. The data will remain in the raw data will remain in the different resources. So also, because we have to start somewhere, we are going to focus on MS data, LCMS or GCMS first. This doesn't mean that we don't consider other data types like imaging mass spectrometry or NMR. This is definitely in the plans for the future, but again, we cannot tackle all problems at the same time, and we have to start somewhere. The idea is that we have common data identifiers for data sets. There will be a consortium agreement. That means that if a resource runs out of funding, the others will agree to take the data, so the data is not lost for the community. There is a common data model, a common submission guidelines and formats, and again, there will be an open portal for the community. So at the moment, these are kind of the first thoughts about submission guidelines. The idea is that the original experimental MS data, the raw data, will be mandatory. Then the process results, both for identification, detection, of quantification, they will be highly recommended, but in the first implementation, they will remain optional, also as data standards develop. There will be experimental and technical metadata, exactly what we are going to be asking for. The information that we are going to be asking for mandatorily is an ongoing discussion. And basically, any other type of relevant information will also be welcome. This is kind of, I personally have a lot of experience in proteomics. I work in proteomics for many years, and this is kind of following some ideas that we did in the MetabolomeXchange project that we were leading. It started like 10 years ago or so. Okay, in context of interoperability, the I of FAIR, Nils Hoffmann will be giving more information about it later, but it's basically highlight here the importance of having data standards that are interoperable between tools and data resources. It is not possible to have 20 different parsers for 20 different data formats and the different versions. So ideally, there should be some community-supported data standards. There are some, for instance, MCML is becoming quite used for mass spectrometry data, but we really need, I think, one format for process results. And this is the work that Nils has started. It started several years ago, and now the effort is restarting again to support new use cases. It's the so-called MCTAP-M data format that, again, Nils is going to introduce later. And other ideas is, for instance, to have a standard way to access mass spectra in open data repositories. So this is called universal spectrum identifiers. We have used them in proteomics for quite some time, and the idea is to standardize them for metabolomics as well in the near future. And of course, the same framework could be used for NMR spectra in the future as well. Also, let me highlight that we don't want to really compete with anybody. This is an open framework that tries to collaborate with as many people as possible. There is no need to duplicate efforts, and we really want to collaborate with people responsible for resources like protocols.io or the lipidomics reporting checklist. Again, Nils is here, or RevMed for compound annotations. Erin is here as well. And really the objective is really to promote more and more the R of FAIR, which is really that the data is reused more and more in the field. There are a lot of nice initiatives, especially in the context of the GNPS team, that they are doing a tremendous work in that perspective, but I think that much more needs to be done there. Really, metabolomics data is underutilized in comparison with other omics data types. And this is really one of the objectives of the project, together with, again, increasing the availability of data in the public domain. So just the final slide to highlight again the main objectives of the consortium: to standardize open data practice in the metabolomics and related fields, including lipidomics and exposomics, for instance. We need to really standardize what is a data set in a public data resource. The idea is also to have a common voice with journals and funders in the context of open data practices, generalize the availability of metabolomics datasets in the public domain, and also, ideally, other data types like spectral libraries, and really to promote increased data reuse for the benefit of the field. So I really want to acknowledge everyone in the MetaboLights team, but also everyone in the Metabolomics Workbench and GNPS teams, and also Nils, who is here, and also the people in the CCRI Hub San Francisco, and especially, of course, to the Sean Zuckerberg initiative for providing the funding for this initiative. And please visit our posters during the conference, and we will be happy to discuss many of these details with you, and also to gather your ideas, because really the whole point is to get feedback from as many people as possible to get things right. Yep, thank you very much.
Okay, questions. We open up. There's 2 microphones. Please, don't be shy. That was all super clear.
Can I ask a question?
Yes, of course.
So,
Nick, can you use the microphone?
is this on? Yeah. So will we still submit data to each of the different data repositories
Yes.
as normal, and then it comes forward? Yeah.
Yeah, so the people will be free to submit the data to the resource of their preference. And this, you know, the idea is that the same kind of assumptions will be used. And then at the end, they will be on the same guidelines. And the idea is that at the end, there is the portal where all public data sets will be integrated. Yeah.
Thank you, very interesting. So does it mean that also after we can have access to all the current and previous data repository reported to all the trees in an integrated fashion to get access to all of them?
So actually, that's a good question. So that has been discussed. And the idea is that, of course, Metabolomics Hub datasets will have kind of, you know, updated requirements, but still in the portal, we plan to enable one option that will allow people also to look for older data sets, what we call, in a way, legacy data sets.
Yeah.
So the portal, by default, will, you know, highlight the data sets that are newer and that, you know, comply with the new requirements, but it will also be possible. The requirements will be slightly different because, of course, legacy data sets are different, and we are still, you know, the data model will be slightly different, but yes, it will be possible to look for older data sets there as well, okay? And the idea, of course, is that as time passes, the legacy data sets kind of will decrease kind of in importance because everyone will be complying with the new guideline. But the important thing is that yes, it will be possible to look for them in the portal.
And similarly for the metadata, I guess.
Yes, sorry.
Similarly for the metadata associated to the.
Yeah, it will be the yes. It's
great initiative. Thank you very much.
Yeah.
Hi. What do you mean specifically by the data being AI ready?
Okay, so that's a good question. I think that there is a workshop tomorrow in the morning that is going to be devoted to that. So data that is AI ready means that, at least in my opinion, that it needs to be well annotated with metadata that is ideally computer readable, and then that the data needs to be generated in a comparable manner. So for instance, maybe for Anna, sorry, Rima is there just behind to highlight this. So it means also that the data needs to be generated in a similar manner. So for instance, for process data, it means that the analysis pipeline that's being used, it needs to be maybe not the same software, but at least the same guidelines in terms of how to decide what is a good compound annotation. So, and then a third component of this is to have suitable data formats for big data. So because for AI approaches, I mean, ideally one needs to have as much data as possible, and this is a bit, additional to the previous two requirements, but ideally is to have the data in a format that is, supported by the people that are doing AI. Okay. So that's what I mean. Annotate it well in a consistent manner, in a way that is computer readable. Data that is being processed in the same way. This is not really needed for the raw mass spectra. And then in suitable data format. This is what it means for me, having data that is AI ready.
Right, thank you.
Mm-hmm.
One important issue is the funding and the continuity of these resources, and we know the funding is always short. So when you said others take it, what does it mean, and what happens to all of this, and who owns what at the end? Who?
Okay, so what I mean is that the consortium needs to have, as you say, funding for data resources is uncertain, but the way it has been done in other fields is that there is some kind of redundancy built in the system. That means that you know, for instance, if, I don't know, I don't want to say any name, but imagine that there is a fourth Metabolomics data repository included in the framework, but you know that data repository gets funding for 3 years, but then the funding is not renewed. It means that the others will agree to take the data on, you know, in this case, MetaboLights, for instance, we will take the data that was submitted to that original data resource and we'll keep it for, you know, for the benefit of everybody, because the problem is that if one resource disappears, then in many cases the data becomes not available anymore. So this happened in the past for us for one proteomics resource that was part of this frame, analogous framework for proteomics, kind of disappeared, but then we took the data and the data wasn't lost. So this is what I mean. Yeah. The way to ensure that there is kind of sustainability, and one of the ways to do it is to build some resilience in the system, and this is what we are trying to do in this way.
We need this resilience, continuity, and a mechanism that is sustainable long-term, and maybe this can come with more participation from metabolomics communities and their funders, right?
Yes.
We need to have a discussion on long-term. Right, that's fantastic.
Yes. Okay.
Thank you very much. Very, very, very cool initiative. Two questions. One is probably unpopular. One is, what's the timeline for this? When do you think that you're going to have something that is working? And the second one is that the amount of submissions is probably going to increase in the future quite a lot. And currently, there's a validation framework. You know, MetaboLights just rolled out number 2. Will this be fully automated in the future? Is that built into this project?
Okay. So 2 questions. So when do we expect to have the first version of the system? So I hope that next year, when we meet in the next Metabolomics Society meeting, I think hopefully we'll have a working version of the framework and even the first data sets there. So I think that's something that it is quite feasible, at least for version 1. Of course, all these frameworks are quite dynamic and still will need some time to stabilize things, but let's put as a deadline for the workshop next year to say that at this point we'll have something that we can show. And the second question is about the increase in data submissions. Yes, so this is something that I think we need to kind of be vigilant about how this can work, because of course we need to have a system that is as automatic as possible, and we are trying to do that. But at the same time, there are some requirements that need to be there. So finding the right balance is kind of the most complicated part of this business. And we are trying our best to find the right balance there. So that's what I think. Yeah. So I think that maybe we can continue the discussion later.
Sorry, I thought it was touchscreen there. Me and technology. Hi everyone, thanks to Tom and Juan for the invitation to speak today. For those of you who don't know me, I'm Warwick Dunn from the University of Liverpool in the UK, and I'm actually here with my MetabolomeX Society hat on rather than my research hat, at least for the next hour and a half here. So I want to talk to you today about what the current landscape is in data reporting standards. Okay, so Juan's talked about being able to submit the data and having that openly available. You need a layer of information, of extra information within that to be able to understand that data and to interpret that data as well. So that's what we mean by the data reporting standards. I've called it for small molecule analysis because what I'm going to talk about today, even though we're in the metabolomics community here, actually we're talking about lipidomics, we're talking about exposomics. The types of techniques we're using, the problems that we have, okay, are very similar across all of these. So it's all focused on small molecules. So I'm going to talk about what data reporting standards are briefly, what the current data reporting standards are, and there are different types of data reporting standards available, okay? And we're not talking about changing any of these significantly, about removing any of these, coming up with new sets of data reporting standards. It's about what's here and being able to use those that are already available within there. And then we're going to talk about what we want to do next and who the stakeholders are within that. So what are data reporting standards? I gave a brief summary, but really these are a way, a description, okay, of the experimental study and of the data that's been collected. So what the study is, how the data's been collected, what the data is within there. Okay, so it's got information about the subjects, the biological materials, the instrument that you've used, etc., all within there. And from that, you can ask different questions. And it's important, within open data, each of us might want to ask different questions within that. So we have to think about that. I'm an analytical chemist, I'm thinking about how can I use it for aspects of that. There might be other biologists who want to use it for aspects of that. So we need to consider all of those within there. So it's standardized reporting, basically. And that's really important. So as we've heard before about being AI ready, making sure that it's standardised, all the information there that's available within there. And if you can imagine across a whole study, there is a lot of information that could be included, but it's actually what needs to be included to be able to interpret that data. And it's not necessarily everything that you've done within there. So we want data reporting standards to feed into the FAIR principles, and we'll hear more about that tomorrow morning in one of the first workshops within there. So really, data reporting standards are standardized reporting frameworks. And again, if this is automated in a way, and there are some that are already semi-automated or automated, that really does help you. And it's you that has to do this, okay? And I will say, first time around, it can be a bit painful, but then once you've done it once, it's very much a copy and paste within that. So what are data reporting standards not? And a lot of people say to me, I don't want to be told how to do my experiment. Well, that's not what data reporting standards are. We're not telling you that you have to have a standardized way that you perform a study, that you collect the data, that you analyze the data, etc. This is just about reporting what you do, okay? And that's really important within the metabolomics community, lipidomics, etc., because we all have different assays. We all think we have the best assays, etc., within there. We don't all have the best assays. There's no perfect assay out there. But we're not trying to standardize how you do your studies and tell you how to do your studies. We're just trying to get you to tell us how you performed that study so that we can reuse that data appropriately. So why do we need data reporting standards? Well, basically, it's an important source. Okay, if you think about all that data that you're submitting, hopefully, all of you, into the data repositories, it's a really important source of new knowledge, okay? But to be able to do that and use that and be able to reuse it, that data, you need to know about the data and how the data's been collected within there. So everything, everything from the start of the experimental design all the way through might be relevant within those reporting standards, and that's what we're thinking about. Whether it's for one experiment or for multiple experiments within there. So we use these, why we need these reporting standards, and we need them standardized, is to make sure that everybody's submitting the right information, the correct information, okay? They're not submitting information that they don't need to do, which takes more time, okay, within there. And it's making sure that data is more accessible and can be more easily interpreted, whether that's by us, whether that's by AI in the future, etc., within there. So it's providing confidence in the data and the conclusions that you're being reported within there. And so, for example, including pooled QC samples and information on how you prepared those pooled QC samples is, for example, one layer that's really important for people to assess the quality of that data within there, and including, obviously, the raw data files for your QC samples as well. So there are a number of different data reporting standards available. I'll go through the main ones, at least in my mind. Apologies if I missed any out, and people that have been involved in that are in the room within there. The first one's in my mind when I was still had hair on the proper part of my head, so this is nearly 20 years ago now. But the Metabolomics Standards Initiative. So all of these were published in 2007. The Metabolomics Society came together at that time, and they published 9 papers on different aspects of the experimental process within there, from the chemical analysis, thinking about the experimental design, the biological samples, how we performed the analysis, etc. within there, the data analysis as well within there. Now, these are now 18 years old. There are aspects missing. There's no discussion about QC, for example, within there, and the use of QC samples. No discussion about imaging, etc. within there. but they have been around for a long time. And actually, I've included the citations for each of these, and you can see that a lot of these aren't being used a lot, okay? And this is what we're seeing within the current data reporting standards. They're not being used routinely within there. We're using the carrot approach at the moment, and they're not being used. So that gives a good example of that. The main ones that are being used are chemical analysis, which is to do with metabolite annotation, mainly, and identification. So coming along more recently, and I'm going in order of date when these were released within here. So Mark Viant, an ex-colleague of mine at the University of Birmingham in the UK, he with a group thinking about more about from the Merit project, thinking about regulatory toxicology and how metabolomics could be used within that. And other omics have had similar sort of best practice and reporting standards within this as well. So this was very much focused on one application area. The MSI standards were across all applications. This is in one application area, and it's definitely worth a read of that. And then along came in 2022 was the lipidomics minimal reporting checklist. There's been a lot of work before that, and I've been involved heavily within this as well. And this is really good. The others aren't online. This is an online portal, so it allows you to complete a virtual checklist as you go through that, and you get a report at the end of that within there. And so that allows you to go through in a semi-automated way to be able to work through that. And I really like it. I think it's a way to go. But that's for lipidomics. So it's not talking about metabolomics. It's focused very much on lipidomics within there. The next one, I think I saw Jennifer in the room somewhere. Jennifer led through MQAC. So MQAC is a Metabolomics Quality Assurance and Quality Control Consortium. So they published reporting standards focused basically on the QA/QC processes that you would apply within a metabolomic study within there. And there's a checklist that you can use to be able to make sure that you're including all the relevant information in there. That was published in 2022 as well. And then more recently in these aren't in order. Sorry, I can't count. This is 2021, sorry about that. But BP4NTA, so these are the best practices for non-targeted analysis. These are based in the US. So these are more away from necessarily not as core as metabolomics and lipidomics, but still thinking about small molecules, including exposomics within there. They have an online tool, a study reporting tool as well, which you download, you complete, and then you can submit with publications, etc., when you're depositing data within there. All of these are different, okay, in different ways, in what the areas they cover, the areas of the process they cover, whether looking at targeted or untargeted data, etc., within there. So overall, we're covering most of the bases within there, but I don't think one reporting standard covers everything within there at the moment. So is now the time to update? Well, I personally think it is. I've taken it on in my role as president of the society to try and get all of these communities together and be able to promote these, make sure that they're usable, for example, with data repositories with what we've heard about Metabolomics Hub within there. And we've seen really, for example, the MSI standards are 17, 18 years old now, and they are not used routinely. Okay, and so there's been a sort of carrot, okay, there within the community. It's not probably working, so there needs to go more to a stick approach that we see in other omics. So when you're submitting data to any proteomics journal, you have to submit your data, and maybe that's the way that we need to go within the metabolomics community within there. But we are working with the multiple stakeholders across the multiple communities within there. We've had one meeting this year. We've broken up into a subgroup to look, compare and contrast across the different reporting standards. And then the next step beyond that will be the application of those and making sure that people are using the right reporting standards, okay, within there. And we're not really going for a harmonized single set of data reporting standards. I think this is quite clear. We don't want to start from square one. There are really good reporting standards out there. We want to make sure that, one, you are using reporting standards and you are using the reporting standards that are most appropriate for you. Okay, whether it's in metabolomics, lipidomics, exposomics, etc., within there. So we're not trying to say that the MSI are the best standards, etc., or the lipidomics are the best standards. We want to learn from all of those and make sure that we can use all of these in the right context within there. So there are a lot of stakeholders, including everybody in this room, okay? But the Metabolomics Society, we've taken it on to try and bring these communities together, and we have done that, and it's great. And all the communities are really engaging within this. So thank you to everybody. I know I can see some familiar faces that are involved in this, so thank you for that. So we do have the Metabolomics Society, we've got the International Lipidomics Society, we've got BP4NTA, we've got MQAC that I mentioned as well. Obviously we've got the data repositories as we're here today, etc. within there, and also including the funders as well. So what are the next steps? Well, we have already brought stakeholders together in January this year. We're naming this initiative PRISM. We've just had a vote across everybody that's involved within this. So this is precise reporting in small molecules. It will be called PRISM 26 or 27 when we finalise this within there, in case we do edits, etc. within the future. We're agreeing to ensure the future use of data reporting standards, and we want that to be standardised within journals, within data repositories, etc. So when you're submitting papers, when you're submitting data to data repositories, you're using these data standards, clearly data reporting standards. We agreed that we're not going to integrate and develop a single set of reporting standards. One, we think that's difficult. Two, that would nullify a lot of excellent work that's already been done within the different communities within there, and we're not here to do that in any way. This is more about signposting, okay, to making sure people are using the appropriate reporting standards within there. And we all want to agree on what the minimum information is. So, for example, for data repositories, we want to make sure whatever community you're coming from, we want to make sure it's the same minimum information that you're reporting within there. And so, as I said, we've got a small subgroup that are working through the different data reporting standards and seeing what's similar, what's different between them, what should be best practice will come from that. And that's it. So thank you very much for listening. I always know it's a difficult question with Steve.
Yeah. So this issue in metabolomics is in fact across the whole of science.
Yep.
Are you in contact at all with Jake Chen in the US? Because NIH has created a U54 program to really facilitate how people can upload their data. You know, some of these sites where you go to load data, it can be quite hard. I know from my lab's experience, just getting the data right. And so what they're trying to do is to standardize the approach of how you upload all NIH data. So I don't know whether you are in contact, but you probably should be.
We have NIH core staff within the consortium. I don't think we have Zach on there, but we'll see each other at the hotel, so write down his name and I'll get him involved.
Okay, thanks.
Thanks.
Thank you for that excellent talk. I was wondering, what are the current hurdles of people using the standards? Because, you know, I'm looking, there's a paper from 2007, right? And we're still having the same discussion about the standards. So what are really the barriers for people using the standards? Is it the standards are not clear, or it's just burdensome to do it? So I'm interested in knowing all the causes for not adopting standards.
Well, okay, so let's do a poll. Okay, don't just trust me. Okay, so please be honest, because this is really important for us. So hands up here if you've submitted one or more data sets into a data repository. That's good to see, yeah, about 50% probably, okay. Hands up if you've followed data standards when you've deposited that data. Okay. For those of you who didn't put your hands up, why didn't you use the data standards? Was it because it was difficult to use those data standards? Put your hand up. Was it extra work? Put your hand up. Everybody's nodding their head, extra work. Yeah, it is extra work. And there was a workshop yesterday and that was a big discussion about it. I will say, yeah, there is extra work in doing that, but you are benefiting the community, the scientific communities at large, by submitting your data and making that data available and making it reusable. It's easy to submit, relatively easy to submit data, but making sure that it's reusable, that's a really important thing within here. So do put that time in. And there was Stefan Newman, I don't know whether he's here yet, was talking about in the discussion yesterday about start that process of depositing and using the reporting standards on day one, not when you've written your paper and then suddenly you realise you have to submit your data within there. So yeah, it is a bit cumbersome. I will say I will agree with that. When you do it the first time, when you do it the second, third, fourth, fifth time, it becomes a lot easier and a lot quicker.
Thanks a lot, Rick. Yeah, this is super important, right? So I agree. I mean, you talked about the carrot and stick approach, and that's an interesting concept, looking at the references and hands up, etc. I mean, one of the key things in other areas is getting the journals on board to make this necessary, right? This is, I think that's a major kind of gate that will be important here. What are the mechanisms that we have as a community to sort of develop that? Because it's very ad hoc and depends on the journal, etc., etc. Given particularly that metabolomics has a very broad reach across many journal types.
Yeah, that's a really good question. So there is evidence that the journals will want to use this as standard reporting practices. We've seen it in other omics. We've discussed already with some journals, and there are some of the higher impact journals, Nature, etc., within there. And actually, within a number of journals as well, MQAC, the MQAC reporting standards for QA/QC, they are standard with some journals already. So they published those a couple of 3 years ago, and they've pushed them through into the journal. So we're early in that process, but there is a path there, and there doesn't seem to be huge hurdles there to be able to do that. I think if the community is saying we need to do it, they will take that on board. Thought I was going to get off then.
So yeah, I agree with the point that probably the stick will have to come in place, right? And one thing I was thinking also related to the first talk is that, for example, my experiences with the BP4NTA, so we have this standard reporting tool, and it's, as you said, quite extensive. So I think it makes sense that a journal asks for that together with your submission. But maybe if there's also a way to do the same kind of thing across the metabolomics hub, so when you submit your data, it has to be this standard reporting tool format.
Yep.
And then either you do, if you don't want to put your data there, you just do it with the Excel. It's basically the standard reporting tool. But if you want to put your data there, then this is already included kind of in the submission of the data to the repositories.
Yeah.
That makes life easier, probably. Yeah.
Yeah, I agree with that, yeah. Thank you.
Hello, can you
Hello?
hear?
Yep.
Okay. Thank you so much and for the talks. It's very nice to see the discussion here. Concerning the previous question that our colleague there asked, there's also another question, and it's a bit, I'm sorry, a bit provocative, which is how safe are us researchers submitting the data that other researchers afterwards are not going to use in a wrong way? And what I mean is like stealing. It's not stealing if you share it, but afterwards they can use it without referencing our data set. And for example, for the journals, I think it was in the first call, like the submission of the paper to a journal, and we put the data set and everything. But I feel like it's not only the standards that we need to follow and everything, but also like us researchers feeling like there's other researchers there that are going to use this data, or other or editors or other reviewers that are going to maybe steal the data or something like this. I know it's a bit provocative, and we all know there's a bit how can I say, afraid of this, but yeah, it's maybe something that, or maybe we can all ask the audience if they also feel like it's something that maybe they are not doing because they fear that the data is not going to be shared.
So hands up, I mean, this is the ethical aspect of using others' research data, which is beyond this workshop, but...
Sorry, I know it's beyond, but I'm sorry, but it's also a bit part of it as well.
How many people wouldn't want to submit their data to a data repository because of that concern?
Good.
That's nice to see.
It's nice, yeah?
Yeah.
Okay.
Okay.
Thank you.
Great, thanks. And I think I'm over time.
You already started mine. Thanks. Perfect. My name is Nils Hoffmann. I'm from the Research Center Jülich in Germany, part of the German Network for Bioinformatics Infrastructure. And I've been involved in quite a number of standardization initiatives in the past for mass spectrometry and also lipidomics. And my background is from computer science, so I'm a bit more on the bioinformatics and applied science side. And I will also try to give an overview into some of these standards and how they can actually be applied, but also talk about some future developments and how they can contribute towards building up this Metabolome hub. So going back to, I think, Rick's talk about a FAIR, let's look a bit more into the interoperability and reusability aspects of research, so the I and R in FAIR. Usually, if your experiment fails, whose fault is it? There are different possibilities for that, so someone probably messed with it, could relate to someone didn't follow best practice guidelines, how to do the things, and how to ensure that everything is done in the right order, for example. The instructions were badly written, someone didn't follow minimal reporting standards, so you cannot really reproduce or recreate the experiment that someone else did. But that also applies to data that has been stored somewhere, of course. So if it hasn't been described in a way that you can actually use it, it's not really helpful. So we need to describe what was done and how, and that helps both in analytical reuse and also computational processing. Last but not least, the computer might have failed to process your data. So that usually comes back to using well-structured and validatable formats to get across this problem. So if you have these formats defined, then you aid processing and interpretation by both humans and computers. And last but not least, that is the basis also to enable large-scale integration and combination of data, and as a basis also for machine learning applications. And I also would like to highlight, coming back to some of the discussions we had earlier, that having the data in these formats that you can then submit is also helpful for your own lab, for example, because you can then use standardized tools and algorithms basically to reanalyze your own data and also do things within your own lab across your old previous studies, for example, that you would otherwise have a hard time doing. One example of these minimal information guidelines was also briefly mentioned already previously is the Metabolomics Minimal Reporting Checklist. This is an online interactive basically wizard that you walk through different stages of your analytical experiment, starting from the overall study design, going to pre-analytics and sample material handling, lipid extraction, this is where it gets really specific towards lipids, the analytical platform, lipid identification and quantification, quality control, method validation, and reporting. A lot of the steps in between are not necessarily tied only to lipidomics, so they might also apply for exposomics, for toxicology, general for small molecule mass spectrometry analysis. And the good thing is this reporting checklist has source code that is also openly available under a very liberal license. So people can actually take this, re-implement it, extend it. And the basic data model also allows you to export the information in a JSON, in a structured JavaScript object notation object. So those of you who program probably know more about that. Last but not least, you also get a citable DOI for your checklist report. So that's, I think, something that also goes back to crediting not only data sets, but also method descriptions. This is something that others can cite, and that also increases, of course, the impact of your own work. So going towards more details about data standards, I would like to go forward talking to you about the UPO-PSI initiative on mass spectrometry data standards, and specifically where we are working towards standards for small molecules in mass spectrometry. To give you a bit of context, so UPO-PSI is part of the Human Proteome Organization. So as the name implies, originally mainly concerned with proteomics, and so they developed data standards for that in the first place, but in the past few years, more and more active work groups have already extended their scope towards small molecules. And some of the working groups, like the QC working group, are, for example, also from the beginning working across the different omics boundaries. We also focus a lot on defining the UPO-PSI MS controlled vocabulary that some of you might have come across if you've worked with MZML in the past, for example, or with other standards that we produce. And there are also lots of interactions with the Metabolomics community already, and also the Lipidomics Standards Initiative and others. PSIMS CV, a controlled vocabulary, is something that allows you to clearly define and communicate concepts about, for example, certain things in your study, like, for example, is the term that you see highlighted down here, the collision energy concept is clearly defined, and so everybody who references this can then agree on the same terminology and definition, so there's usually only a small amount of misunderstanding or ambiguity left, and hopefully none at all. This is an open controlled vocabulary, so if you find anything in there that's missing from your point of view, you can easily contribute via GitHub and also get in contact. And usually the turnaround time for integrating new CV terms is between 1 and a few weeks. Some of the standards that we are working on concerning small molecules currently are MZTab for metabolomics, that I will go into more detail on later. The universal spectral identifier that was also briefly mentioned, that allows you to cross-link spectral information into repositories going back to the raw data files and identifying which spectrum they originated from, if you, for example, have identification information. SDRF, currently for proteomics, which tries to establish a sample description metadata format, but is also currently being extended towards metabolomics as well, which allows us to actually also cater for multi-omics experiment setups, where we don't want to redefine the same sample description for all of the different omics, but we can basically just create one SDRF, or maybe have SDRF core, and then for proteomics and metabolomics, and then reuse that in the different specialized reporting formats. On top, we have MZSpecLib for spectral library information, MZPAF, which is currently really geared towards proteomics, but might also be extended towards metabolomics in the future to annotate individual peaks in mass spectra. And last but not least, MZQC. So there are also new working groups booting up at the moment, for example, on AI, and we are just now reinvigorating the MZTab for metabolomics working group. So if we group these standards across different axes within the UPPSI, we can split them into the categories of sample metadata for SDRF, for MS data with MZML and USI in a sense, but that's also a cross-cutting concern for other of these formats. Identification, which is for small molecules, primarily MZTab for metabolomics at the moment, and for final results as well. And there we also have the spectral library format and MZPAF. And cutting across these different aspects, we have MZQC, which allows you to express QC-related information on individual runs or also group of runs. I would guess that most of you have probably come across MZML before. This is quite an old format nowadays. That was published in 2011 and is very well supported also from the vendor side. So the mass spec vendors also support MS Convert, that then can use native vendor libraries to create the MZML files containing a lot of the metadata we need from the raw machines, basically. And this is also closely tied toward, to the controlled vocabulary of the UPPS IMS. At the moment, there is also an effort to define a new, more efficient format that is better suited for technological advances that have been made in the past few years, and also tries to be more efficient towards large-scale data. So for MZTab-M, we need some sort of motivation. So most of you probably have seen something like this before in the past. So just an Excel table with some identities with your sample information, molecule names, maybe some information about treatment and concentrations that you have. And this is in some form or way usually the basis of what you would report and also submit. But of course, this has some issues, so there's not really a defined vocabulary here that is available. That's just your own internal vocabulary, basically, that might work within your group. So everything here is not really standardized. So the aim of MZTab-M is actually to make these kinds of reports more machine readable, better annotated, semantically annotated, and also include identification evidence and feature data from your experiments. So why do we need to define what we are talking about specifically? We already briefly mentioned the CV, the UPPSI MS CV. We need to exactly know what we are talking about if we want to share data, because otherwise you might define thing A as something and I might define it as something different, and we don't really understand what we are talking about. So that's why we need to make sure we talk about the same things. MZTab for metabolomics is a data format that tries to group this kind of information for small molecules into 4 different levels of information. This can be represented in a tabular way, so all of these 4 sections are basically tables within one large file. But we also can serialize into JSON, for example, nowadays. This captures metadata information about your study variables, assays, MS runs, etc. And the top level, then on the summary level, this is your typical report table that you would submit, for example, as a supplementary information for your publication, containing all of your identified metabolites, for example, across your different assays with the final quantities, for example, assigned, and identities linking back to, for example, external databases. This is then supplemented by the MS feature and quantitation level, which actually contains the raw information from your extracted mass spectra. And then finally, the identification evidence level, which allows you to express identification information from database hits, for example, or other kinds of identification methods. So this is modular. So one example for where we apply controlled vocabulary terms here is how do we report ID confidence. The MSI, for example, defined this four-level scheme in 2007, which allows you to report what is known about a molecule and how ambiguous the identifications are in principle. There are other schemes to report ID confidence. This is just one example here on the right-hand side, which expands this original scheme for high-resolution mass spectrometry, but there are more available as well. And by assigning defined controlled vocabulary terms for each of these, we are able to actually communicate to someone who's reading the file what kind of system were you using to annotate and also to express your confidence in your identification. So we are currently in the process of updating the format. We found out in the past few years when using it with some of the different implementations that we need more flexibility concerning the number of levels that we can report on, to also allow to include this, for example, in XCMS workflows to successively build up the information from basic information, for example, starting at the feature level, then adding the metadata and summary level information later, and then complement that with the identification evidence. There's also potential to collaborate in the biohackathon later this year to actually try out some of these ideas in implementations and also do cross-conversions between different formats of UPO-PSI, so if you're interested, just get in contact afterwards. So, very short final words on MZQC. So, you all know that if you're running machines with chromatography, mass spectrometry, or maybe even other technologies, you have lots of sources of variability and errors in your workflow. Those need to be captured somehow. Most of you probably run specific QC samples, for example, or have other means to control this variability or at least measure it in some way. The MZQC format is intended to report on these kind of QC metrics. For example, if you have batch effects that you can observe with the PCA of your samples or other QC metrics as well. These are all defined as controlled vocabulary parameters again, and so this is very easy to extend this format to your own application and use it as a basis, for example, for your own LIMS system. Final words on the data storage and sharing. So basically to interconnect everything with this data hub and also to interconnect tools, we need adapters. And MZTab for metabolomics, for example, is one such adapter. So it's a format that contains at least a minimal set of information that you need to report, but also different communities can define different levels of minimum metadata for this file format. So it's pretty flexible and extendable. And in that sense can also cater to different communities. So it doesn't really only apply to small molecules or metabolomics or to lipidomics only or exposomics only. Everybody can basically use it as a basis. And the next steps here are basically to also extend this with other formats to make it more modular, to be able to cross-link to MZQC, for example, to MZSpecLib, or other formats outside of the scope of UPO-PSI-MS, if someone of you is involved in that. And yeah, last but not least, that makes your data more fair and also not only reusable by others, but also by yourself. So with that, I'm at the end of my talk, and thanks a lot.
Do you think some kind of integration between MZTab and MSG common data model might be? And
MSG?
Yeah.
It depends. We have to see whether that is possible to link it out or maybe map one to the other. That's definitely a good question. And if you want to talk more about that in detail, I think it's easier to talk one-to-one on that specifically.
So one of the concerns I've had of late, going back looking at the data we store, is that the data is stored in a coded form by the mass spectrometers. How that data comes out into a form that we can use in different programs is a point of variation. I found that if you take a raw set of data and convert it into different formats and then look at it in one of our software, we end up often with different answers because peaks get split, zeros get inserted in different places where they shouldn't be. Can we do something to address this? Because I think if you put bad data down, then you're going to get bad data interpretation.
Yeah, I get the problem. So I think one of the main factors also with the Metabolome Hub is to also store the raw data always. So you always have the possibility to go back to the vendor format. But that of course means we need somehow support by the vendors to get out the data in a way that doesn't lead to artifacts and doesn't increase the variability. And I think that's something that we cannot solve alone. So that's really something where we need support and also define testing for that.
Do you see the possibility for MZTab-M to sort of ease upload to these repositories in terms of a direct interface, making it, I mean, I think that's a big barrier to entry for the community.
Yeah, definitely. So the idea is, so we already have a lot of tool developers on board. And so MZMind, for example, MSDI used to support it. I think we'll also support it in the future again. So if you're using these tools, then you can create an MZTab-M at the end of your workflow. Also with XDMS, we're in the process of doing that. And then you can use MZTab-N basically as a lingua franca, so to say, to do the submission. And then you would have these different community-specific validation profiles that then allow you to say, okay, this conforms to the minimal information required by Metabolomics Society, by the Exposomics community, or by the Lipidomics Society, and so it can go into this or that repository. Also telling you what's still missing in the validation. So I think that's really the important part. And yeah, we're actually, if you want to check out my poster number 2000B, I think, I'm showing a use case of doing this kind of data integration based on MZTab-M, just the metadata and summary level information, showing how that can actually help you compare across different quantitative lipidomic studies to better understand how, for example, lipid concentration ranges vary over these different organisms or also over different analytical methods, which is really insightful.
Excellent. What day is that poster? What day is your poster?
I have no clue right now. I could check it. Sorry.
So as you can see, I am here representing the Exposomics community. Essentially, this is largely introductory because we're kind of joining the game relatively late as a community via the aspect of how technology is developed and being used for the broad measurement of environmental chemicals in samples. So a little bit of background is I'm based at RECETOX, Masaryk University in the Czech Republic, and then also I essentially had a research group on human chemical exposome, but also operate a laboratory. I'm on the analytical side, and we also have a subunit on compute. But also, I am here largely on behalf of research infrastructure, which is IRENE, which is then the Environmental Exposure Assessment Research Infrastructure. So essentially, as I said, my lab is just a classical mass spec lab. I'm on the analytical side, and then also just here, just to say, we do operate open science practices. We are pushing this. We have like all our SOPs and everything uploaded. We have a Zenodo community. The compute side does all open coding. There is the GitHub, and I also, if people noticed, raised my hand where we do deposit into repositories. And essentially, we linked via the fact that sometimes standards currently haven't yet been extended to incorporate exposomics fully, and we essentially link to update certain things as we don't want to redo. So essentially just the research infrastructure is a background that's quite simple, but essentially a research infrastructure is designed to support excellent scientific research. So essentially it provides and develops services, it gives technical support and training, and also it's there to enhance innovation. So this can include technology development, and it's also co-creation, so it's open to industry and commercial partners. There's a landscape analysis within the EU, but various other countries have national infrastructure roadmaps. They're highly strategized. They're essentially countrywide level, so it's often ministerial, but they cover, at least in the EU, various domains across all sciences and aspects, and also digital infrastructures and technology infrastructures. So essentially, IRENE is just an open access, distributed research infrastructure. So this means that IRENE has nodes in various countries. There are other single-sited infrastructures. But essentially, we joined on the EU landscape to support research on exposome, and then a major component being chemical exposure, which is then our link into this community. And then also population health studies. Essentially, we have a large current background for human exposome, not the wider exposome community currently, but it is designed to be inclusive. It can branch out and extend. So essentially, it has currently 50 institutions in 22 countries. It is going global. You will notice that we have, for example, the UK, USA, and Australia already present, so it isn't EU-centric. A point here is also that via certain funding mechanisms, for example, funding currently under certain calls from the Commission, funding can also be outside of Europe. So we can have partners, for example, where we can fund Canada if they wished, for certain work, for example. And essentially, again, this is highly just to support transnational access. So it's cross-country collaborations, cross-country development, just to support scientific research. Within IRENE, the major services that are relevant here is then the chemical exposure profiling, but we also then have biological response profiling. So essentially, if we're measuring environmental chemicals and relating them to health, we also then conduit that via mediators. Mediators essentially then endogenous responses. So this covers not only metabolomics, but also, for example, protein and all layers of omics. This falls under then exposure characterization, then it links into hazard and risk assessment, but also we host then environmental data, so that's like geospatial data or environmental maps. Within ours being human exposome, we have human data. This is largely just human population cohorts, because there are other infrastructures which host clinical cohorts, but essentially prospective cohorts that do not have clinical outcomes largely weren't represented, so we have a portion of that. And at the end, because it's highly multidisciplinary, integration, data processing and tools and platforms, a large component comes onto mass spectrometry again. So essentially, we have many corporations, but if anyone is doing anything within the remit of the exposome, please join, for example, the International Human Exposome Network, which we directly link through IRENE and then to the US comparable enabling coordinating center for exposome research, which is Nexus Exposomics in the US. And these then have already linked IRENE, IHAN, Nexus. The stakeholder community was shared. Obviously, that stakeholder community is particularly EU-US. However, they are wishing to go global, and on that sense, there is an Exposome Moonshot Forum, which then is widening out for more global stakeholder participation to kind of give a meeting point and a community for the wider research. Another project partnership that I will talk about in the next slides is PARC, which is a 400 million co-fund pan-European project on next-generation chemical risk assessment. This is just an example of where we interact that is across all EU member states. And obviously, being the European research infrastructure level, we cooperate with other research infrastructures, whether they be European research infrastructures like BBMRI ERIC or Share ERIC, or intergovernmental organizations like EMBL, and then EBI as a component of the EMBL. So it's just large corporation. So here for Nexus, which I said is the complementary US coordinating center to enable exposome research, we directly link essentially to the wider unit, but obviously exposome is heavy. But I then cooperate directly with the ChemBio Hub led by Tom Metz and Crystal Pollett. The Nexus Hub in the US is pan-NIH institutional funding, and essentially within their proposal, I pulled out this figure here where essentially they proposed to generate Exposomics Workbench. They'd linked out to Metabolomics Workbench and essentially proposed and kind of push that, and we got in touch being like, oh, this would be much easier if things were harmonized. Please do not duplicate activities everywhere, essentially. PARC also, as I said, it's very large, but it has quite a lot of MS-relevant activities. Within PARC, it's focused largely on monitoring. So then there are labs doing human biomonitoring and also environmental monitoring. Currently, PARC comprises of 270 mass spectrometry labs. These largely, many are accredited or feeding regulation, but this just basically means that there's a lot of people using mass spec technology. There's a large capacity, and slowly they're branching out into doing more full-scan analysis, or also going into what they are deeming biological markers of effect, which then essentially also covers metabolomics in some way. And the challenge here, we reach out, is that they sometimes are relatively unaware and haven't looked around to the disciplines largely on the basis of different terminology, and we are attempting to make these bridges. For example, within the innovative analytical methods, this includes full-scan screening, non-targeted profiling, and how that would inform regulation. We led the recommendations to agencies in the Commission of how that should inform next generation chemical risk assessment, but this means that we can directly communicate with, for example, ECHA, EEA. So this is a link in ministerial policy and decision making. There's other deliverables currently where we have interceded to branch out because they also do a lot of work on their data infrastructures, making things fair, tools, and cataloging resources, metadata, what they wish to report, just because they're simply coming from a different perspective, but often using the same technology and capturing quite a lot of the same data by the end. So essentially, as I said, we're just bridging then genotype, exotype as a different representation of phenotype. We do not wish to encapsulate everything. We want to extend, work with people, build out, and just modify. Obviously, on the right-hand side, you can see that there's a lot of contention between how to demarcate between a chemical exposome and metabolome when something is obviously internalized and biotransformed. And we do not actually wish to have this contention of people in the field segregating and siloing their work, which is then great for the small molecule usage rather than having segregated terms. So essentially the benefit is that we have the same primary technology, largely mass spectrometry. However, differences are that we inherently expect much more sparse detection of environmental chemicals and their metabolized products. We also have a much greater abundance variability, largely on the basis that we have kind of passive environmental chemical exposures, which are on average 3 log orders of magnitude lower than most typical endogenous metabolites, but also we have, for example, active exposures. So we, for example, will take medication, which then spans huge ingested amounts. So it means that these kind of anthropogenic chemicals are typically a very, very broad range of abundance. Also, for environmental chemicals, there is a lack of reference chemicals, and so there is a lack of reference spectra. What I mean by this is we will never acquire the reference spectra for many chemicals, largely due to legislation where we cannot access them because they are proprietary products in industrial processes. Particularly within Europe, this is protected in legislation, so they will not be reference spectra for many analytes in the public domain at any point. And that causes that we often have to interact with a lot of chemoinformatics, a lot of in silico work to promote that. So we're kind of doing things from a different side. This is because we're inherently asking different research questions, and then that also has knock-on effects where we would have importance and place emphasis on our different metadata fields for use. And we also have very different requirements on QA/QC, which then is where we do not wish to completely be isolated, span off, and as a research infrastructure, we have a large community asking for support on this research, and I do not wish to repeat the decades of work that has been gone on in other fields, so this is where we branched out. However, in a contribution, obviously, for us to interpret our data depends on metabolomics knowledge, particularly as the interfacing as the biological representation of phenotype. However, that's also vice versa. So where you see variability, that's typically because there has been a change in environment, and that is a very good sharing of knowledge, framework standards. There's also a large proportion of researchers moving into chemical exposomics in a broad manner screening, so now moving from doing more quantitative, targeted, selective ion acquisitions to full scan acquisitions, and they're moving in from a very heavy regulatory background, so there's a lot of QA/QC proficiency, metrics for assessment, and quantification, qualification of how would you assess full scan data being involved in this field, which the actual how would you interpret true positive, false positive rates, etc., could be something contributed into metabolomics because that is somewhat lacking. Also, there are the close linkages to decision makers and policy feeding of data, which then helps to advance societal impacts. Also, the use of environmental variables advances just gene-environment interaction studies, and this, again, from our side in a clinical ecosystem research sense, can actually move from single case control setups to multi-case setups in a much more simplified manner. And then also we connect to other large-scale research infrastructures that operate mass spectrometry. This includes, for example, AITRIS, which is the translational research infrastructure, which is clinical, Instruct, which is structural biology, that also have large-scale labs, and all these infrastructures essentially are designed to support people to use them and provide services. So going there. Just as an evidence of particularly what we have contributed from my lab, I have to say again, I'm the analytical side. I just have a compute subunit run by Helga Hecht, who isn't here today. But essentially we've interacted and cooperated where we've provided multiple tools to the Galaxy ecosystem. We have 20 open code repositories. We create whole workflows to process our spectrometry data and put them public. You will see that we participated in the Research Data Alliance on multi-omics metadata standards. We participate and cooperate with the Norman network of laboratories, the BP for NTA I also sit on. I go and attend the Quark meetings, and again, the Elixir platform, so the European Life Sciences Data Resources. It's another research infrastructure. We contribute and attempt to get into the communities just to be kind of a place, a meeting point, sharing knowledge, not repeating things. And then inherently here with EMBL-EBI, and also we basically want to deposit a lot of data somewhere, and within Europe we obviously chose MetaboLights, and that is where we bridged out very early on to EBI. So it's largely that we do capacity building, and we work with Workflow Metabolomics in France, for example, for our tools, but we also contribute to ontologies such as EDAM. We've contributed to BioTools. You'll see just there's many, many logos where I can say that we are contributing. It's not that I'm advocating for this without personally doing it. We do contribute, essentially. Then I just have many acknowledgements, in particular EMBL-EBI, because as I said, we reached out to them very early on because we do not wish to do anything that they have done excellently before. So thanks, Thomas, Juan, and obviously this is wider than just EBI, but also EMBL, where Amy Foreman, for a lot of other aspects of exposome research where it intersects, all partners of IRENE, IHAN Nexus as the linkage, and then in particular Jana Klanover, who coordinates IRENE, and Helga Hecht, as I mentioned, who is the Bowel Compute subunit head, and then various national funding projects and things, but it's more people than I can place, essentially. Thanks. Questions? Great.
Not one question? Okay, we'll warm you up with some prompts. So one activity I personally wanted to gain from this workshop is I put together just a little spreadsheet. So maybe if you're interested in this topic, just add your name to that spreadsheet and kind of interest that you're thinking about, that you're interested in, and just see if there is enough critical maths yet and what the topics that people are interested in. I'll leave the QR code in all the prompts as well. So yeah, let's see if we can get something rolling. I've just switched sides, but the QR code will still be present. Okay, so we've got 2 themes, I think, with the time frame left that we want you guys to give us input on, and we want to hear your insights. One of them is around the priorities. So I want you guys to start thinking about what are the top priorities right now? What deserves our focus? If we accomplish one thing, what should it be? And are there any small wins or intermediate steps that can be achieved within the next year? And while you're thinking on these prompts, again, feel free to fill out the spreadsheet, but also we'll ask our panel to contribute, and I also just want to welcome 2 new people to the panel as well. So Brian DeFleece and David Wishart, if you'd like to come and join the panel too. I would like to start from the audience, but if not, I will go to the panel. Anyone have anything they would like to share? No one has any priorities? You guys just complaining it's hard to deposit to the public domain? Okay, maybe we can start off. Who should we pick? Any preference? Elliott, would you like to share some thoughts on this opinion?
I don't think it's the easiest to start with, in the sense that we have many needs for extension, purely on the basis of if it's metadata, we have a very different perspective. If it's on the true standards of the actual formats per se, we contribute standards of the library's representation, we're involved. That isn't particularly difficult. Our challenge is more the extension, metadata representations, ontologies, terminologies, where a controlled vocabulary is very simplistic within the technology domain, because the technology is the technology that everything uses. When you expand out of variables that are not technology-specific variables, those variables then just are using different words, essentially, in different communities just by inherently, and they will never use the same words. It will be that you have to map and you have to define what they really mean. It's a lot of communication where even if they are using the same words, they view the value of the word differently. So ours is very much on a terminology communication basis, priorities, where we just want, from my side, it would be to have greater engagement and representation of essentially the exposomics community here, and greater representation of metabolomics researchers, people in lipidomics, people doing anything to do with small molecules or mass spectrometry in general, also in the exposomics meetings. At the moment, we're at the stage where it's communication.
Yeah. Oh, yeah, sorry.
Just a general comment, perhaps we can use the time tomorrow at 8 o'clock for those who will attend, is to think about big data to knowledge. It's wonderful that we are organizing all of this data, to be able to use it, but then what's the ultimate goal? The ultimate goal over 20 years, you know, as a metabolomics community, we have made great progress. We do not have the home runs yet. Yes, that says, listen, as a community, we can identify a mechanism of disease, try to treat, maybe we are getting closer. Yes, we can have an impact on something, right? Let's say human diseases, one thing. So from big data to knowledge, from thinking about the metabolome as the net interaction of the chemical exposome, the gut microbiome, the diet, yes, the socioeconomic, all of this as the parallel to the genome. How do we put all of this together to say, listen, the metabolome is giving us the readout for these net influences on health. I hope tomorrow we'll be able to tackle some of this as a beginning, but organizing the data, using it, is clearly a first step towards that goal. Yeah.
First of all, thank you very much for the great talks. I would have two questions indeed. When we think about data repositories and about data quality, we oftentimes think about MS1 stability, MS2 fragmentation patterns. We think about annotation levels. But in many metabolomics datasets, there is another dimension of chromatography. Are we also considering standardizing reporting of gradients, solvents, columns, making that even mandatory when uploading our data? And the second question, building up on this, could we even envision introducing retention time standards more broadly?
Yeah, I think I can answer a bit to that. So within Elixir, the European bioinformatics infrastructure that Elliott mentioned, we currently have an implementation study running that also Thomas is included in. And one part of the work package is actually to look at current reporting standards for chromatography, if there are any, and to revise and also define what we want to actually report there to make it reproducible and reusable for others. If you look at how MetaboLights is doing that right now, they have the method sections in the submission part, and that's basically free text. So there's some sort of, I would say, mild agreement how to write this down, because the method sections are usually rather technical. So that's also something that could be interesting in the future, to look into trained machine learning models to actually extract this kind of information from the textual description to help with the annotation. But that's something that we are actually currently working on. So we're in the moment also creating a survey to send around and share with people in different communities to learn about how they would report chromatography conditions, for example. And that's going to also feed into these activities then afterwards. The second part of the question was?
Retention time standards.
Yeah, it's a really good question. Obviously, everybody's using different assays, different columns, etc. So there isn't a single, at least at the moment, there isn't a single retention time library out there that will work for everything. But as Nils just said, if we've got all this information, and I know there's people within the community within Europe are working on this and being able to transfer across different assays, etc. as well. So including it now, it will be useful in the future.
Maybe I can pick on you, David. Do you have any priorities for the community?
So I think there's perhaps a couple. One, I think it's really important to be able to work with the software developers, the ones working in MS Style or MZMine or XCMS and others, to make sure that the formats that they're producing, the data that they're capturing, is in a format that we could just basically, click once and upload to the repository. So they could make it a lot easier for everyone, because what we're really demanding of people now is to publish twice. First, you have to write your paper, and then because no one believes you, you have to publish a second time into a repository. And that's really onerous. and frankly, it's unfair. And I think if you look at other fields where they have, DNA sequencing, every file that comes out of a sequencer, whether it's Illumina or any other PacBio, Oxford, Millipore, Nanopore, whatever, those are in a format that you can just click and upload. They're in FASTQ, they're in image files, they're in FASTA. People don't have to put everything that they've ever done into that file uploading process. And I think there's probably more than 24 different pipelines that have been developed. We mentioned the 3 or 4 most popular, but then, probably if we did a review in this room, we'd find another dozen that other people are using or even writing themselves. But if we could get people to agree that this is a format and this is the information that their pipeline should capture, then it should just be 1 click, 1 minute upload. And I think that should be a major priority.
Yeah, that's a great point, and actually something that the Metabolomics Hub will try to help with. We will, advocate for these standard formats, MZTab-M, for example. I don't know if anyone else from the audience have some favorite ones they want to shout out. And then we will work with, the software developers. So we'll work with Robin from MZMind to get this implemented so it's easy for you guys to export. And then once you export it, we're working with, MetaboLights will hopefully have a little converter that you can convert into MetaboLights and metadata, everything.
Oh. I think she was first.
Yeah.
Now, just thinking about what they've just mentioned, that XCMS or MSDI or MZMind people should try to kind of develop this format. Should that be the way, or should we maybe also put some pressure on the vendors that if you do an LC method, there is already a format that is the same that you just upload to MetaboLights, it's readable, and we can all work with that? That's an idea.
Comments from the panel?
Yeah, I would basically like to add to that. So getting vendors on board, I think, is really, really crucial, because not everybody uses open source tools or is not even allowed to do that, because for regulatory reasons, they need to have approved software. So having the vendors on board there really helps. And the other part would be, so within Elixir, we are trying to also increase the adoption by basically including these activities in biohackathon projects, trying to get the developers on board. But those are localized activities in Europe. So not all developers are in Europe, of course. So I think that could also be a really good help from CZI, basically, or from the BioHub, to support these adoption hackathons across the world to help other tool developers to actually get these standards on board and get them implemented, because then also the distribution within the field is getting much wider, and we get the critical mass basically to also create the format in as wide as possible.
Yeah, certainly. I think we also, need to agree on what this final digestible AI-ready format is, right? Is it a matrix of here's my metabolites and here's the confidence I have in the annotations that I made, and maybe some free text that describes the methods? What level of depth do we want on that sort of unified metabolomics hub data set compared to, I think, Juan, you mentioned that the raw data will still be hosted on the individual repositories, and there'll be sort of a composite on the hub. So deciding on exactly what that combined format is will help us figure out how to create that in a more automated way.
One issue as we look forward to think about is in every grant we write, in every funding we seek, there's got to be a component for data sharing. If that is the future, and it is, we are there. I give you one example from an experience of NIH. The NIA, David knows the initiative there. They wanted to adopt open science as a model across the NIH campus, and they were serious about it. Yes, not only the data, but the metadata, a topic for maybe tomorrow, because the metadata and the harmonization of the metadata is as important probably as this biochemical data that we are trying to put in. But the adoption that was made was every grant that gets funded needs a component to formulate this data, get it ready for public sharing, and hence the AD Knowledge Portal, for example, became a recipient. Every grant has a portion, X number of hundreds of thousands of dollars goes there. Is there a vision? If you guys are adopting this beautiful big vision for the field, let us think about what is a parallel that can bring you more sustainability of funding so everybody benefits. Let's think about models.
Yeah, great thoughts, great comments.
So maybe I can comment on that a little bit. So this is really not kind of an issue of metabolomics. It's an issue of all kind of biological, well, even not only biological disciplines that are producing large amounts of data. So in an ideal world, and I completely agree with you, there should be a kind of a stable funding mechanism. What you have explained, it would be ideal that that both the data sharing activities and the resources are kind of funded in a sustainable manner. There is a lot of lobbying going behind the scenes on this. Large institutions like NCBI, EBI have been advocating for this for a while. There is now an initiative called Global Alliance for Resources, Core Data Resources. That is also, so I think this is not a solution that will, come tomorrow. It's like, a small sum of many parts that need to be kind of put together somehow. And as a community, we can only advocate for this. So, yeah, I completely agree with your vision. I'm just saying that, and there is a lot of activities going in this direction. But
the society can play an important role, and maybe
also the societies, yes.
Vision forward. From 20 years when we started the society, vision forward is a proactive, something that is more in line with vision forward and bigger needs and goals.
Maybe that's a good segue into my next topic, consortium building.
One more comment on the funding perspective. So in Germany, we have lots of local initiatives at universities now to establish data stewards and data stewardship offices. And they are usually experts in certain application domains as well. And they are exactly there and funded to help people create data in a way that it can be submitted to repositories, for example. And I'm not sure whether everybody does that, every country does that, probably not. But those are really important initiatives that also need to be risen to in awareness to policymakers, of course. So in Germany, we have the National Infrastructure for Data Science, in principle, NFDI. And that also tries to harmonize that across the different disciplines. And that's also, of course, very helpful to have such a kind of initiative. But if societies can get behind these things and also drive this on a policy basis, I think we can move this forward to also get the funding from the state side, basically.
So thinking of that, so what initiatives, activities, programs should we be engaged with? What roles could other resources play here? For example, HMDB, Mass Bank. Are we missing opportunities? I think we are. And what steps can be taken, as Reem has suggested, to maximize this future growth and sustainability? I know these are deep prompts now. I spent a lot of time on these prompts.
Yeah, thank you. So I'm just thinking, I'm not sure if this is what you were prompting me on, but I guess just reflecting a bit on the last comments as well. Just think about spheres of influence here and the sort of duplication that that's come up in terms of multi-omics. We're often doing many omics actually, and bring those together. Particularly one of the things we want to do in metabolomics. So, I'm sure many of us in the room have actually sort of faced with the situation where we're uploading data from transcriptomics, proteomics, and metabolomics, and they're all very separate at the moment, and the communities are at different levels of integration in terms of repositories. So I guess thinking future growth and sustainability, how we link together those aspects, I think are quite important.
Yeah, great. I think someone mentioned the multi-omics working group. Elliott, was that you? you want to comment on that?
So there is under the RDA like working groups for multi-omics metadata standards, MOMSI, where essentially it's pointing researchers towards the different standards in the different domain, and then of those, which ones could then be integrated. I'd also have to mention that EMBL-EBI have many resources, for example, biostudies, where if you deposit in different repositories, different data types, it then does kind of provide you a linkage and a home where it's not directly like integrated data, but it is cataloging at least where everything goes. Currently in this field, there isn't, in my opinion, enough data. Like, people are not depositing data. It's very difficult then to push integration when you're collaborating with domains that inherently are larger. For example, proteomics is larger, but also genomics in particular is huge. And then typically many of these initiatives, as before, they have to be kind of nationally driven from a ministerial or policy level. By virtue of the scientists that turn up, just by probability of the domain, the people in contact then with national ministries and representatives, by probability are from those fields and are not representing domains that are smaller. In Irene in particular, we have very good links at a national level. We have similar goings on as Germany with data stewardship, where we as a research infrastructure, our end goal is to be able to integrate. For exposomics, my focus is the environmental chemicals, but that has no relationship, as Rima was saying, to actual knowledge of how it influences health of people or the environment. We do wish to push this, so that is one benefit, that that community, by virtue, has policy links. It's what we build, but it's representation and presence, turning up and communicating to those people.
Yeah, maybe I can add that from the EBI perspective here. So yeah, we are lucky that we have all the different data types, you know, but it requires resources and a forum to get the people together. So we have a little multi-omics working group at EBI where we try to get, you know, the genomics, the ENA, nucleotide, come sit with me for a minute. We're lucky with the proteomics as well, the PRIDE, come sit with me for a minute. And yeah, there's a lot of efforts we can learn from that, and again, to avoid the deduplication, especially around the sample, project, study level metadata. What's fair, they've kind of already got a lot of thought and consideration around that, but really it takes, everyone's super busy, so it takes resources, and then, you know, spend some time, talk to one another, and then you can start to figure out the problems. You know, I have the same sample, I probably need a unique persistent identifier for this that then I can link to the resources. We will mark up this slightly betterly, so these kind of issues, yeah, but it takes resources, time.
Hi, I just had a few comments. So in terms of resources, one thing that could be really interesting is to reinforce the creation of reference materials, because we have this wonderful NIST material that is actually expanding. We have, I think from Avanti, in terms of lipidomics, there is like this yeast total, yeast polar, metabolic, and this is really nice, but I think we really don't have enough. And having, for example, like a cell polar extract, some kind of very standardized, that people just could put into their cohorts, into their sample sets, that we could just, when we upload data to repositories, that would be easily identified, we could trace them back. And then second comment would be in terms of instrumental parameters, to actually try to retrieve as much as possible from what we researchers think the information is. Can be the raw data, that all the instrument parameters should be there, depending on how you acquire, but most of it would be there. Sometimes depending how you hyphenate for example, your LC with your mass spec, sometimes you are, if they are not married well because two different vendors, then you don't have the LC parameters or not enough granularity. But when we actually publish, we do the effort, at least some of us do the effort to put everything in terms of like Excel format, like all the columns and so on, but how, I think it would be really easy to just try to provide a link where we could upload this kind of material so that the repositories could mine it and that it could be already filled in as much as possible without us just trying to copy paste from the Excel sheet to the things. So,
yeah, great. Any comments from the panel?
I can comment on the reference materials, where one, I've been involved in various initiatives to generate various types of reference materials. The generation of, in particular, reference matrix materials or like certified, standardized matrices is typically not economically viable. The generation of just pure standard mixtures also at scale largely becomes not economically viable once they're labeled or overly diverse. The benefit in the environmental community is largely due to the regulatory need. Typically these standards and reference materials, obviously by design, then they're pushed via agencies like NIST, or also in Europe, there's then the Joint Research Centre of the Commission. So for example, within the PARC project I mentioned, we are then in direct contact with the JRC of what reference materials the JRC is essentially going to create, and then because there is a need on a regulatory level, they have to create. For metabolomics, I'd probably say that it's actually worthwhile going and talking to people in very much the regulatory clinical domain, reporting diagnostic assays, doing quantitative work, bringing in the like full scan procedures, linking it more to labs which particularly do report under clinical accreditations diagnostics, and explain to them the benefit for future discovery of, for example, just biomarkers, prognostic, diagnostic, like, because they're the ones that then have to mediate through the policy aspects, because it tends to not be economically viable, and it's often that policy drives what is available. The same as when you contact NIST. NIST, for example, with their materials, they wish to provide materials, it's just economically they can't. And it's, I'd say maybe going through clinical for that connection again could push that.
Yeah, about the reference materials, I think you also, when doing untargeted work, need to be careful how you're processing and aligning your data, as you could put your thumb on the scale, so to speak, and have an annotation from your reference material that then is maybe gap-filled, which I think is quite standard in data processing algorithms, where all of a sudden you're seeing annotations that are actually from your reference material and not from your samples themselves. I was actually thinking of this yesterday. Rick mentioned that a lot of data on repositories doesn't have QC samples. And we wrestle with that, where maybe we run 100 samples of various types, and when we look at the data, we say, you know, actually the question we're asking is only these 50 samples, and we've pooled 100 samples, so our QC is made of 100 samples, but we're actually not doing anything with, you know, certain bacteria that are in that data, so we align without the QC samples. Well, we have the QC samples to make sure that the system worked, and like that's another, I think, issue with how we're representing our data and reporting our data is to be careful not to bias it.
People in the audience, you are part of any standard efforts? Put your hands up. Anyone's part of any standard efforts out there? Any standards efforts? MQuack, we have some people at MQuack, yeah? Okay, good. So now you know all these new all these new activities, initiatives you should go and find out and join. Maybe we could take from the panel, anyone wants to take about maximum future growth and sustainability? What steps? Maybe Rick. There was a comment about, maybe Rick, I can put you on the spot, about the society's role here.
I do agree with Rima. We need to be forward-thinking. We definitely are forward-thinking within the Metabolomics Society, within the board and beyond that. You know, but we are a society, we're not a funder. You know, we're here to support the society, the community within that, in every way that we can within that. So reporting standards is obviously one of those. And we can be the voice for the community within that, and the long-term voice of the community. But certainly we don't have millions of pounds to give out. Definitely we don't have that to support.
I mean, maybe I could comment, but in terms of sustainability of data resources, I think everyone in the community can do a little, in the sense that, you know, if in many cases, some resources may have been given for granted, and especially now with the situation in the US and the you know, the funding struggles that the NIH is going through, it turns out that you know, many of these resources that are funded by NIH are really in big trouble. So so everything that the community can do to support them, it would be very kind of, I mean, everyone can do a little bit. So for instance, if the resource you are using, you know, asks you to fill up a survey, maybe, I don't know, just talking about something that is happening now at the EBI, and you say there that, you know, you use this resource, you use it for this, and this is really important for you, that can actually help a lot. And there is no other way, I mean, there is no way to guarantee, just to make it clear, that, you know, in 5 or 10 years, there will be funding to support all data resources. I mean, we hope there will be, because at the moment this is how it has worked. But yeah, there's no guarantee. I mean, political priorities can change, and this is this is the situation right now. So what we can do, I think, is to build resilience in the system a little bit. This is what we are trying to do in this Metabolomics Hub approach. But yes, just think that, you know, resources that you may be using now, they shouldn't be taken for granted. That because all the resources need funds to exist. Yeah.
No, I think these are really good points. At some level, worrying about what's happening at the federal or higher levels, pulling away from support. And then at that point, maybe you have to think about how do you do it locally. And one of the areas I think we tend to neglect or ignore is the fact that many of us are at universities or at research institutions. And across the world, there are thousands or tens of thousands of those. All of those research institutions actually have libraries, and they're still sustained, but it's sort of, we have to start wondering, and I asked this question before, but how many of us have actually walked into a university library in the last 10 years? And almost no one puts up their hands. And yet, a given university will spend millions of euros or dollars each year sustaining these libraries. They'll also sustain archival collections. And the archival collections used to be focused on collecting, you know, rocks and shells and other things. And they spend a lot of money maintaining those as well. Somehow the university systems has completely lost track of the need of data and the data that scientists generate, and how we tend to use this electronic stuff almost exclusively. We don't use the books, we don't use the archival collections. And so if there's a way of essentially convincing the universities to direct some of their essentially core budget to the support of data and data archives at a universal level, then we could have the sustainable support for these databases. But it seems that there's a disconnect. Universities intrinsically always allocate millions of euros every year to things that no one uses, but they're not willing to put that same amount towards the data resources that all of us generate and that all of us need. And so we need to talk to our universities and convince them about the need for data sustainability.
Oh yeah, sorry.
Yes, so I sort of sat and thought about this a few years ago. I gave a talk on how we store data. And of course, all of us want to have data immediately available. Now, the expansion of the data that's being laid down is going to exceed all resources. I mean, my chair bought me a new PC to do metabolomics data, and had a terabyte of memory, and I filled it up in about 6 weeks. So I can't keep that data. And so the question is how to more efficiently store data, particularly archival data. So do we need to keep it on hard drives? I mean, that's what we all like, but it's actually very expensive compared to what you might otherwise do. And so you can go into the cloud and put stuff into MagTape at about 1/10 of the cost of putting data down on a hard drive. So is that something when we talk about archiving and the expense of data, is that something that we should consider?
Maybe I can comment on that a little bit. So, I mean, there is usually a correlation between on how accessible you want your data to be and how expensive the system to store the data is. So, I mean, I think the solution for this is to have different tiers, no? So maybe the data that was generated, I don't know, in the last year, you are very interested on that, and then you store that on the most expensive type of, storage. But the data that was generated 5 years ago, or I don't know, even more, it is very rare that you will come back to it, and then you store it in, but you still need to do it, you still need to have it, and then you store that on, on tape, which is, again, much cheaper. But of course, for that, there needs to be a strategy in each kind of university or research institute to be able to implement this. And of course, it could be done in a similar way across different. The only thing that complicates things a little bit is if you are dealing with sensitive human data. And if you are dealing with human sensitive data, then because of all the ethical concerns and everything, then everything becomes much more work and much more also expensive in parallel. But having 2 systems for open data and sensitive data and having these different tiers, I think this is the way forward to handle the situation.
Thank you.
Any final comments? If not, just we would really like to engage with you more. We have posters here. We're trying to build a little consortium, so do approach us throughout the workshop. Give us some feedback. We have Owen here from Metabolomics Workbench. Yasin's representing GNPS Massive. So yeah, and just thank you so much for your participation in the workshop, and yeah, have a great time.